The topic of Chapter 7 is the nervous system. The nervous system can be divided into the central nervous system, or CNS. This consists of the brain and spinal cord, and the PNS, or peripheral nervous system, which is the cranial and spinal nerves. Nervous tissue is composed of two types of cells: neurons, which conduct impulses. They're said to be excitable, but they generally can't divide. So there are some exceptions to this. There are parts of your brain where you do have adult stem cells that can, in fact, become neurons. But in general, it's true that you're born with a certain number of neurons, and that's all you have for the rest of your life. Glial cells, also called neuroglia, are support cells. They do not conduct impulses, therefore they're not excitable cells, but they are able to divide. In table 7.1 from your book, we can see a couple of these things we've talked about already. The central nervous system, or CNS, is the brain and spinal cord. The peripheral nervous system, or PNS, is nerves, ganglia, and nerve plexus. All of these are outside of the CNS. Now, neurons can be generally grouped into three categories. One is the sensory neuron. These are referred to as afferent neurons also. So those are sort of interchangeable terms. Afferent refers to the idea that impulses or a signal is moving from the periphery towards the central nervous system. So neurons that transmit impulses from sensory receptors to the CNS. So those two terms are interchangeable. A sensory neuron is also an afferent neuron. Motor neurons are another big class of neurons. These are efferent neurons. So these are neurons that transmit impulses from the CNS to an effector organ, for example, a muscle. It could also be a gland. And so again, you have the term efferent. Efferent refers to information flowing out from the central nervous system towards the periphery. So motor neurons are efferent neurons. And so again, those two terms are often used interchangeably. Nerves are a collection of axons in the peripheral nervous system. Now, some nerves are going to be primarily motor, others are going to be primarily sensory, and others are mixed. They're going to be, mix of the two, sensory and motor. There are different types of motor nerves. There are somatic motor nerves. These stimulate the contraction of skeletal muscle. Autonomic motor nerves stimulate the contraction, or they inhibit the contraction of smooth muscle and cardiac muscle, and they can also stimulate glandular secretion. Ganglia is, ganglion, singular, ganglia, plural. This refers to groups of neuron cell bodies located outside of the CNS. The same thing, but within the CNS, is a nucleus. So if you have a grouping of cell bodies, often neurons that have a similar function are located close together, that would be considered a nucleus. Tract is sort of the equivalent of a nerve, but within the CNS. And I almost neglected to mention the term interneuron. So you have sensory neurons and motor neurons, and all other neurons within your body or within your nervous system are called interneurons. So the vast majority of neurons in your brain are in fact interneurons. And these are located entirely within the CNS. So neurons are the structural and functional units of the nervous system. They have some general functions. They can respond to chemical and physical stimuli. So this would be things like sensory receptors in your skin. They can conduct electrochemical impulses, also called action potentials, which we'll talk about more later. They release chemical regulators. These can be neurotransmitters, or they could be neurohormones. Finally, they also enable perception of sensory stimuli, learning and memory, control muscles and glands. Most cannot divide, but they are able to undergo some level of repair. So neurons vary very widely in terms of their anatomy, but there are some general trends we can see. They generally will all have a cell body with a nucleus. various organelles such as endoplasmic reticulum, mitochondria, things like that. They cluster in groups called nuclei within the CNS, and they cluster in ganglia in the PNS. So one common thing that most neurons have are dendrites. These receive impulses and conduct a graded impulse towards the cell body. And this graded impulse, we'll talk about this more later, it's integrated at the where the spikes are generated near the cell body at the axon hillock. Most neurons have axons. These conduct action potentials away from the cell body. In this diagram from your book, you can see the structure of two kinds of neurons. The first one here, you can see has a cell body with a nucleus, and it has dendrites. Coming off the cell body there's an axon hillock. This leads to what's called the initial segment of the axon. And from there, the axon may bifurcate. So you'll have what are called axon collaterals. And these collaterals themselves may bifurcate another time. So axons are actually often quite complicated with many branching points and so forth. In this particular case, we can see here the endings of the axon are called axon terminals. They're also called synaptic boutons. Bouton means button in French. Here's another type of neuron. So this would be more typical of a sensory neuron in your body. You have dendrites out in the periphery, say in your skin. These would act as receptors for, say, touch or pressure. An action potential would be generated within the dendrite. The action potentials would move past the cell body and into the axon and finally into the central nervous system. The cell body in a typical sensory neuron is actually often exterior in the peripheral nervous system in a ganglion. Axons can vary in length from a few millimeters to a meter in length. They're connected to the cell body by the axon hillock, where the action potentials are generated. So it's a little complicated. So the tapering part is called the axon hillock, where the cell body tapers. And then after that, you have the initial segment. That's the first part of the axon that has a sort of standard diameter that will continue as the axon moves further away from the cell body. As I said earlier, the axon can divide many times. It can bifurcate, meaning it splits in two. And each branch is then called an axon collateral. The axons in vertebrates are often covered with myelin, with open spots called nodes of Ranvier. And we'll talk more about that in future slides in a second. Because axons are so large in terms of length, they can be, as I said, they could have an axon with a length of a meter. They need to transport materials. And this is called axonal transport. These are active processes that move things like organelles and proteins from the cell body to the axon terminals. There's basically two types of transport. One is fast transport, axonal transport. This moves membranous vesicles, which often may contain things such as a neurotransmitter. And these vesicular cargoes can move relatively fast, 200 to 400 millimeters per day. There's a second kind of movement, which is a slow component, and this involves the movement of microfilaments, microtubules, and proteins, and this is much slower, with a rate of about 8 millimeters per day. There are two types of axonal transport. There's anterior grade transport, which is from the cell body to the dendrites or to the axon. So it's essentially moving away from the cell body. The molecular motor for fast axonal transport is kinesin. And here's a picture of kinesin walking along a microtubule. You can look us up on YouTube. You'll see interesting videos. What you have here is some sort of vesicle, which may contain something like neurotransmitter or contain something else, possibly. And then it's being dragged along by the kinesin molecule, which actually has 2 feet, essentially, that walk along microtubule. So this is kinesin for anterograde transport, to transport materials in the other direction, retrograde transport towards the cell body from either the dendrites or axon. There's another molecular motor called dynein. Here's a picture from your book of some of the parts of a neuron. You have the cell body here with its nucleus, dendrites, axonal hillock, which tapers into the initial segment. So around this area of the axon hillock initial segment would be the spike initiating zone where action potentials are generated. Here's an axon. In this particular case, we have a neuron that has a myelinated axon. So myelin is a kind of a wrapping that typically has a whitish color because it consists of a lot of phospholipid membrane wrapped around and around the axon. And so this acts as an insulator, an electrical insulator. And in this case, we're talking, this particular neuron is in the periphery of, in the PNS, or peripheral nervous system. So we have a Schwann cell here with its cell nucleus, and you can see it actually is a fairly normal cell. Out here, it has cytoplasm and so forth, but it's towards the interior of the cell, where as it wraps the axon, it has this interesting myelin sheath, in which there's very little cytoplasm. And you know, from one Schwann cell to the next, there's a gap, and this is called the node of Ranvier. And we'll talk more about the significance of this node later. So we've already talked about some of these things. There's a functional classification of neurons based on the direction of impulses. So sensory neurons conduct impulses from the sensory receptors to the CNS, so they're considered afferent neurons. Motor neurons conduct impulses from the CNS to target organs, which could be muscles or glands, and these are considered to be efferent neurons. And then, as I said, interneurons are the vast majority of neurons in your body, and these are located completely within the CNS, and they integrate functions of the nervous system. Motor neurons can be divided into two major types. Somatic motor neurons, these are responsible for reflexes and voluntary control of skeletal muscle. And then there's autonomic motor neurons. These innervate involuntary targets such as smooth muscle, cardiac muscle, and glands. And there's basically two divisions to the autonomic nervous system: the sympathetic division. This is activated during emergency situations or fight or flight situations. And there's the parasympathetic division. These are involved in rest and digest functions. And we often talk about these as, the sort of extremes, fight or flight versus rest and digest. But in fact, all the time, both of these systems are in fact activated. It's just the degree to which the sympathetic and parasympathetic systems are activated. So most of the time, you're not in a completely fight or flight situation or a completely rest and digest situation. You have a little bit of both going on. Okay, this slide shows quite nicely some of the things we've been talking about. We have this central nervous system here represented with the spinal cord. You can see there's a couple things to see here. First of all, here's the sensory neuron. There are receptors, perhaps in your skin here, that would detect mechanical disturbance, such as pressure or touch. An action potential would be generated within the dendrite at some point here, where I'm pointing. The action potential would travel in past the cell body and into the CNS, or central nervous system, where it may release neurotransmitter onto an interneuron, as shown here. Now, somatic motor neurons have their cell bodies within the CNS, so they're considered to be central nervous system neurons. Despite the fact that, in fact, the majority of the cell membrane is actually out in the periphery. So here's a very long axon. It goes out to a skeletal muscle. And again, you'd have the synaptic boutons or axon terminals in which a neurotransmitter would be released. So somatic motor neurons are, in fact, because the cell body is in the CNS, would be considered to be central nervous system neurons. The autonomic nervous system is a little bit different. It's designed differently. You'll have two neurons, one with the cell body within the CNS, projects outward in an axon. It then releases neurotransmitter onto a second autonomic motor neuron, which is located within an autonomic ganglion. And then this autonomic motor neuron sends an axon to the actual smooth muscle, cardiac muscle, or glands that it innervates or connects to. So neurons are often classified based on their structure. One way to do this is based on the number of processes that extend from the cell body. In a pseudo-unipolar cell, you have a short process that branches to form a T to form two longer processes. An example of this would be the sensory neurons that we were talking about earlier. Another kind of neuron is the bipolar neuron. These have two processes, one on either end. An example of this is found in the retina, and I'll show a picture of this in a second. And then finally, there are also multipolar neurons. These have several dendrites, usually one axon that departs from the cell body. This axon, of course, can split many times into collaterals, but there's just one axon that comes off initially. And this is a fairly common type of neuron. Here are pictures of the types of neurons we've been talking about. So a typical sensory neuron in your body that has its cell body in the dorsal root ganglion, which is located, a ganglion that's quite located relatively close to the spinal cord. It would have a dendrite, which would have some sort of receptors. Again, we could imagine a mechanoreceptor branches here. So when there's some sort of pressure, say, on the skin, there would be an action potential generated within the dendritic branch about here, where I'm pointing to. The action potential would travel in past the cell body and into the central nervous system. And then here are the axon terminals where we could release neurotransmitter and activate the next neuron. Within your eye, as I mentioned earlier, you have bipolar neurons. So these would receive neurotransmitter from another neuron. This would excite these dendrites a bit. We'll talk more about how this happens exactly. But an action potential would then be generated within the dendrite, travel through the cell body and out the other end along an axon. And then finally, neurotransmitter would be released at these axon terminals. So that would be bipolar. Finally, we have multipolar neurons. These have a cell body with many dendrites. So there's synaptic input that comes in to these dendrites. You have the axon hillock and initial segment. So somewhere in this region is the spike initiating zone where an action potential is generated. And then it travels away from the cell body typically towards the axon terminals. And you can see here we have bifurcations and finally many axon terminals. As I mentioned earlier, you can classify neurons. Nerves are, again, bundles of axons located outside the CNS. In fact, most are composed of both sensory and motor neurons, and these are called mixed nerves. But you have some that are just sensory nerves, and that would be a sensory axons within it, and so that would be considered a sensory nerve. Others, occasionally, you'll have nerves consisting of only motor axons, and those would be considered motor nerves. Some of the cranial nerves have sensory fibers only. Within the central nervous system, we don't call as neurons project from one area to another. We refer to those as nerves anymore. We refer to those as tracts. So glial cells are extremely important, or these are also called neuroglia. These are non-excitable cells. They don't conduct action potentials, but they have a very strong supporting role for neurons. Two types we can talk about that are quite important. You have, for example, in the peripheral nervous system, Schwann cells. These are also called neurolemocytes. These form myelin sheaths around peripheral axons. And then there are also satellite cells. These are also called ganglionic gliocytes. These support cell bodies within the ganglia of the PNS. Within the central nervous system, there are four main classes of glial cells. There are oligodendrocytes. These form myelin sheaths around the axons of the CNS neurons. So the term oligo has to do with a few. So they have a few processes off their bodies. And I'll show a picture of that in a second. Another class are microglia. These migrate around the CNS tissue. They actually move, and they have sort of amoeboid movement. They phagocytize foreign and degenerated material. Astrocytes have many more branches. Astro refers to star, so they're star-like, and the fact they have many processes. These regulate the external environment of neurons. We'll talk a lot more about astrocytes. They're very important. Ependymal cells are another type of glial cell. These line the ventricles within the brain. The ventricles are fluid-filled spaces, and the ependymal cells are involved in secretion of cerebrospinal fluid. This is the special fluid that lies within the ventricles of the brain, as well as within the certain regions of the spinal cord. This picture shows nicely some of the types of neurons we've been talking about. For example, here's an astrocyte. You can see it has many different branches. And you can actually see some of the functions it has. You'll see that it has these perivascular feet. So this is a capillary within the brain. And so it's involved in, these feet are involved in taking up nutrients from the capillaries. And you can see that the same neuron will actually send foot processes to an actual neuron. And so in this way, it can actually help transfer nutrients from the blood to neurons. And they also play a role in the actual formation of something called the blood-brain barrier. So they're important for that also, with these foot processes directly on the capillaries. And we'll talk a lot more about astrocytes in a second. Here's an oligodendrocyte. Note that it has fewer branches. Here it has a foot process on this neuron. Here it has a foot process on another neuron. Here it's making a myelin sheath. Note the node of Ranvier here. And so oligodendrocytes are the equivalent of Schwann cells, but within the CNS. So Schwann cells are found only in the PNS, whereas oligodendrocytes are found only within the CNS. Now here's a microglial cell. So it's involved in immune function and attacking foreign bodies such as bacteria. They also are involved in breakdown of cellular fragments, for example, from a dying cell. And then here you can see ependymal cells. So here we have cerebrospinal fluid. This is presumably something like a ventricle within the brain. And so here the ependymal cells they're very important in the sense that they line the ventricles. Table 7.2 is a nice review of some of the things we've been talking about. We've mentioned Schwann cells. They're located only in the peripheral nervous system. They're also called neurolemmacytes. They produce myelin sheaths around the myelinated axons in the PNS. They surround all PNS axons, myelinated and unmyelinated, to form a neurolemmal sheath. So it's often assumed when you think of Schwann cells, you think of myelin, but in fact, there are Schwann cells that also protect non-myelinated cells. So they simply don't form the multiple layers of myelin, but they are there. They are present around axons, even if they're not myelinating the axon. Again, satellite cells support functions of neurons within sensory and autonomic ganglion. So they would help with supporting metabolism, things like that. Oligodendrocytes, are found within the CNS. They form myelin sheaths around central axons, producing white matter in the CNS. So this is an interesting point. So whenever you have myelin, remember it's many layers of phospholipid bilayer wrapped around and around. And so that's essentially going to be white in appearance. because it's essentially a type of insulation that's essentially lipid, right? So it's similar to fat, and you can think of fat as being white, right? And so any place where you have a lot of axons that are myelinated, you'll refer to that as white matter. Areas where you have fewer axons, more cell bodies, or perhaps the axons aren't myelinated, that would be referred to as gray matter because it has a darker appearance. Microglia, these phagocytized pathogens, and they will also just remove cellular debris in the CNS. For example, if a cell undergoes apoptosis or programmed cell death, it may leave behind some cellular debris that would need to be cleaned up. Astrocytes are extremely important. They're found all over the CNS. They cover capillaries in the CNS and help to induce those foot processes. We saw a picture of them earlier. They actually help to induce the formation of the blood-brain barrier. These interact metabolically with neurons and modify the extracellular environment of neurons. We'll talk a lot more about astrocytes later. Ependymal cells, they form this epithelial lining of the brain cavities, the ventricles. Also within the spinal cord, you have a fluid-filled area called the central canal, so they would be found there also. Ependymal cells also cover the tufts of capillaries to form choroid plexuses. These are structures within the ventricles, within the central canal of the spinal cord, where cerebrospinal fluid is actually produced. So ependymal cells are very much involved in that. Okay, so the myelin sheath in the PNS is formed by Schwann cells. And the axons of all PNS neurons are surrounded by Schwann cells. So even if they're not myelinated, they would still have Schwann cells. These Schwann cells wrap around the axon to form the myelin sheath in the PNS. And then the gaps, as we mentioned earlier, would be called nodes of Ranvier. And we'll talk more about the function of these nodes of Ranvier. This picture illustrates a little bit better what goes on. So you have, you can imagine here is an axon, and then you have a Schwann cell, and it's essentially just wrapping around and around the axon many times to form the myelin sheath. And then outside of the myelin sheath, you have the cell nucleus and the other, the endoplasmic reticulum, mitochondria, and so forth. And then you have this area of cytoplasm, which is fairly normal, which is called the neurolemma. So not all cells have myelin. So small axons with a diameter of 2 microns or less are often unmyelinated. So these would conduct action potentials relatively slowly. We'll talk about that more about that later. Even unmyelinated axons in the PNS have a neurolemma, but lack the multiple wrappings of the Schwann cell plasma membrane. So they would just simply lack the wrapping that would form the myelin, but they would have a neurolemma in the Schwann cell, or Schwann cells, plural. And this is something we'll talk about more later, but myelinated axons will conduct impulses much more rapidly than unmyelinated axons. In this picture, you can see a myelinated axon. You can see the myelin sheath here. You can still see the Schwann cell cytoplasm. The nucleus is not visible, but you can see it. It's outside of this myelin sheath. There's a fairly normal-looking cell, and that would be the neurolemma there. And as a comparison, here we have an unmyelinated axon. So it has some sort of surrounding cell, which is presumably a Schwann cell, but you see that there aren't the multiple wrappings of myelin sheath. And here it even says this is the Schwann cell cytoplasm. So this is presumably Schwann cell over here also. In contrast to the PNS and the CNS, the myelin sheath is produced by oligodendrocytes. One oligodendrocyte will extend branches to various axons and wrap different sections of these axons. So these oligodendrocytes produce myelin sheath, but they do not produce a neural lemma the way the Schwann cells do. I've already mentioned this, but so when you have myelin sheath in the CNS, these are going to give these tissues that have lots of axons a white color. So that's referred to as white matter. Gray matter is cell bodies and dendrites, which lack myelin sheaths. So an example of this would be within the spinal cord, you'll see the sort of butterfly-like shape of gray matter. And so that's mostly cell bodies and dendrites, and they're going to lack myelin sheaths there. Whereas around this butterfly shape, you'll have white matter. And that's because there are many axons traveling longitudinally from, say, the lower spine up towards the brain, and from the brain descending down towards the lower spine. And so that's going to be white matter surrounding the gray matter. This is just a picture of what I've been talking about. You have an oligodendrocyte. It's wrapping multiple axons. And you can see the nodes of Ranvier, these regions in which there is no myelin sheath. And again, we'll talk more about the function of those nodes a bit later. Okay, as I mentioned, so myelin is very important for proper functioning of the nervous system. The wrapping essentially leads to the ability of axons to transmit nerve impulses at a much higher velocity than would be possible otherwise. Unfortunately, there are some demyelinated diseases which will lead to things like muscle weakness. For example, there's Guillain-Barré syndrome. T cells of the immune system attack the myelin sheaths of the PNS. This produces rapid onset of symptoms that include muscle weakness. So essentially, the nerve signals are not traveling from the central nervous system to the periphery, and this leads to this problem of muscle weakness. Another example of a demyelinating disease is multiple sclerosis, or MS. This is produced when there's an autoimmune attack by T lymphocytes, causing lymphocytes and monocyte-derived macrophages to enter the brain and target myelin sheaths, causing demyelination. So again, this will have sort of similar effects. There'll be problems with communication, in which action potentials are often not able to travel along the axon efficiently. So there may be a decrease in velocity, and if the disease progresses, it's possible that the action potentials may not be transmitted at all.